Hi everyone, my name is Parleen Pandher and I'm a second year master's student with Dr. Gray at UNBC. One way that we expend energy is through thermogenesis, and brown adipose tissue is the key thermogenic organ. We are interested in the regulation of thermogenesis across different levels of the central nervous system in response to cold stress, and we hypothesize that the neuroendocrine factor POCAP is expressed in the sympathetic nerves innervating brown adipose tissue. Gene expression analysis through qPCR is able to assess differential expression with high sensitivity and specificity, and reference genes are required for the normalization of data to account for technical variation in nucleic acid synthesis and loading. As a first step, it was challenging to complete dissections and extractions for our tissues as there was no published method to help guide our work. We achieved high quality RNA, but due to the small sample size, it was of low yield. There was a gap in the literature for recommendations of an optimal combination of reference genes to use for normalization in the sympathetic nervous system tissues, and so we decided to test multiple reference genes on our samples and to share our findings. We acclimated mice at cold and thermoneutrality for 4 weeks, isolated the adrenal gland, superior cervical, and stellate ganglia as shown in figure 1 here. RNA was then extracted from dissected tissues, cDNA synthesized, and gene analysis performed using qPCR. As a positive control for the dissection protocol, we tested tyrosine hydroxylase, and then tyrosine hydroxylase positive tissues were carried forward for reference gene analysis, listed in Table 1 here. In Table 2, we show the average CT along with the cDNA dilution used for the genes tested on our 3 tissues of interest. There's high consistency in CT values for the same gene among the tissues. Additionally, we are showing the individual genome M value for each gene for each tissue as calculated by the Cubase Plus program. In Figure 2, we are looking at the relative mRNA expression for each reference gene for the adrenal gland, superior cervical, and stellate ganglia. Each graph here shows the highest genome M value with 5 gene analysis, and then once again with 3 gene analysis using the genes with the lowest M values. Table 3 summarizes recommendations for reference genes for the 3 sympathetic tissues tested. For all tissues, we were able to recommend genes with a genome M value of less than 1, except for the female adrenal gland where our genome M is 1.138. This work will be beneficial for the study of sympathetic nervous system tissues, allowing for a streamlined approach to analysis, using less sample volumes and saving on time and costs. Further, this work will be used to detect POCAP receptor expression in these sympathetic nervous system tissues when exposed to cold stress, enhancing our understanding of POCAP's role in the sympathetic regulation of adipose tissue function in energy metabolism.